I'm going to be talking about the variation in structure and function of reef fish assemblages among distinct coral habitats. So, as you know, species composition is changing across ecosystems due to multiple stresses. This is causing the loss of some species with introductions of others. However, a recent global study done by Maria Darnellos and colleagues showed us that rather than a net loss of species at globally across biomes, what we're actually seeing is a consistent trend in community turnover of species. This is starting to be recognized as one of the most pressing but largely unrecognized biodiversity crises of our time. So one way that this is manifesting is there's a differential susceptibility of organisms to disturbances such as climate change. This is causing actually non-random species turnover, which is affecting habitats as well as the distribution of species that associate with them. Therefore, there's a growing concern for how these altered ecosystems will function. Some studies that have been done have to look at the influence of community composition on ecosystem function across different ecosystems have shown that it can affect habitat provisioning, primary productivity, nitrogen cycling, detrital decomposition, pollination, as well as some recent work by Tessa Hempson that you may have seen in her exit seminar, where she showed that trophic energy flow is also affected by differential community composition. The focus of my talk is going to be on the composition of foundation species such as corals that create habitats and have knock-on effects on the structure and function of ecosystems. So on coral reefs, climate change and a range of local pervasive stresses are changing the composition of coral assemblages. And I've just pulled up a few examples here on the left from around the world where shifts have been documented. For example, in Mexico here, Lorenzo Alvarez Felipe and colleagues documented shifts towards more weedy species of Agaricea and Porites. In Kenya, Tim McClanahan and others documented shifts towards dominance of Porites and Favids. This is occurring because of the differential susceptibilities of coral taxa to disturbances such as thermal anomalies and large storms. That causes non-random species losses and shifting dominance patterns towards those more stress-tolerant and or weedy species of corals. Now, whilst the outcome of these shifts is likely to vary by location, altered assemblages are predicted to persist into the future. And this is likely to have implications not only for coral cover, but also for the structure that they provide to other reef-associated organisms. Oh, I was meant to show you that, sorry. So, whilst this is actually increasingly occurring, we don't yet know what the implications will be for reef function. We know that corals are structurally and morphologically diverse taxa, and as assemblages shift from more diverse assemblages to more monospecific assemblages, this is likely to have implications for the provision of habitats as the structure changes. It's also likely to have an influence on the structure of reef fish assemblages, and in particular, I'm going to focus on the functional structure of reef fish assemblages. It's likely to influence critical ecosystem functions, and also what remains unknown is how resilient these different configurations will be to ongoing disturbances. So with that, the aim of my thesis was to influence the role of coral species composition on the structure, function, and resilience of reef fish assemblages. I broke this down into 4 distinct chapters. The first one, I looked at how coral species composition influences the cross-scale structural complexity of coral habitats. The second chapter looks at how the functional structure of reef fish assemblages varies among taxonomically distinct coral habitats. The third chapter takes advantage of a really unfortunate bleaching event to see how pre-disturbance coral species composition can influence the susceptibility of reef fish assemblages to a systemic disturbance. And finally, I looked at whether herbivory varies among different coral habitats. So to do this, I did all of my research up at the beautiful Lizard Island, which most of you will be familiar with, in the northern section of the Great Barrier Reef. At Lizard Island, in the lagoon and up the leeward side of the island, there's actually a really fantastic natural experimental setup with reefs occurring with dominance of different coral taxa, and these are the habitats that I compared for all of my studies. So we've got branching and massive parietes, Postulopora, mostly Postulopora damacornis, staghorn acropora, stuffed coral, and some mixed coral assemblages. And I had replicate sites for each one, and they've all got very similar depth and exposure. I also compared some low coral cover habitats. So just to jump right into chapter 1, asking how does coral species composition influence the structural complexity of coral habitats across scales? Well, the structural complexity of the benthos can be highly variable. And this study by Lorenzo Alvarez Felipe showed that whilst there is a positive relationship between coral cover and rugosity or structural complexity, this relationship is really determined by the identity of the dominant coral species in those habitats. For example, Montastria habitats are more complex with more coral than these parietes reefs. So we know that structural complexity is really important for the organisation of organisms, particularly reef fishes, and also for ecological resilience. But one scale of structural complexity is not necessarily synonymous with another scale, for example, within colonies or across colonies. And this is important because animals will use their environment at scales relative to their body size in refuge selection. So what I wanted to look at is the influence of coral species composition on this cross-scale habitat structure. So to do this, I compared 5 different habitats which I've listed here, and you'll see this in each of the method slides, so you can see which ones I'm referring to. And I measured structural complexity using these wheels where they're run over the reef of a 10-meter transect length, where the smaller wheels will fit into smaller nooks and crannies on the reef and therefore measure a larger contour distance than the larger wheels, which will largely just bounce over the surface. Oh, and I looked at 5 different scales ranging from 4 centimetres up to about 64 centimetres, and I meant to bring the wheels in with me to show you, but I forgot. So I also looked at quantifying the structural complexity at these different scales of different colonies to try to tease out where the influence of colony size on habitat structural complexity. And I used a range of mixed effects linear models and some model selection to look at the influence of coral cover on structural complexity as well. So what did we find? Well, just to pull up the smallest scale first, what you see here is the habitats listed along the x-axis and the contour distance traveled by this small wheel along the y-axis. And you can see that there are differences indicated by the different letters here. You can see that there are differences in fine scale structural complexity among habitats, as you might expect. If we compare that with a larger scale, you can see that actually the relative differences among habitats really depended on the scale that we were looking at. If I pull up all of them, you can see that actually the relationship between the relationship for structural complexity among habitats really varies at every single scale. The greatest difference, again, as you might expect, was at the smallest scale. Well, what was interesting was that the massive parietes and branching parietes provided the most complex structures at that scale. If we look at how structural complexity varies within habitats, what I have here is actually for example, I'm going to pull up the rest in a minute. We've got branching parietes here. We've got the different scales that I looked at across the bottom, the contour distance traveled by each of those wheels along the y-axis, and the letters again indicate where there were significant differences in the distance traveled by the wheels, and the bars here are really just for illustrative purposes, so you can really clearly see where there were breaks. So in branching parietes, there were 4 of these distinct breaks, indicating that structure is available at more scales. If we compare that with the other habitats, you can see that actually it's different in every habitat. Again, interestingly, massive parietes has these 4 breaks, but they're different to those in branching parietes, and that is reduced to 2 distinct structures here in soft coral habitats, and down to a really homogeneous, flat habitat, which you might expect for the low coral cover. At the colony scale, I guess what I wanted to show you here, what was most striking, was not only is of course, the structural complexity different among habitats, but the relationship between structural complexity and colony size was really strongly linear. You can see by these R-square values. I've indicated this here because, of course, there's a departure here as the smallest wheel fitted into all of the individual branches of those branching parietes colonies. So just to sum up what we found in this chapter, structural complexity of coral habitats varied over these multiple scales, and it was underscored by coral species composition. And it wasn't, in fact, a simple function of the total coral cover at each habitat. The relative complexity changed with scale, with fine scale structural complexity varying the most. And it's this scale that is most vulnerable to disturbance, but arguably this scale that is most important to a number of small-bodied reef fishes. So habitats provided structure at some scales and not others, and this has important implications for cross-scale resilience. And so as we're seeing more of these novel coral assemblages, they're likely to create these distinct reef structures, and providing differential refuges for more mobile organisms. This has implications for the distribution of those organisms, which leads me on to chapter 2. So I looked at how the functional structure of reef fish assemblages varies among these distinct coral habitats. So what do these shifts mean for fishes? Well, there's a huge body of work that has really quantified the impact of a loss of coral habitats on reef fish assemblages. And they almost consistently show marked changes in reef fish assemblage structure. They're with declines in abundance and species diversity, changed body size distributions, and alterations to the trophic structure of those assemblages. We also know that at the species level, the use of different coral species varies among fish species, and as well as at different ontogenetic stages of fishes. At a community level, some work by Vanessa and colleagues showed that coral diversity can also influence reef fish assemblage structure. And of course, we know that from a huge amount of work that's been done by Nick and others, that habitat structural complexity influences both the taxonomic and trophic fish assemblage structure. But what we don't yet know is the influence of coral species shifts on fish assemblage structure and function. So there's an emerging interest in using functional approaches to try to determine the impact of these changes in communities on the functioning of ecosystems. So within this context, species are considered in terms of the roles that they contribute to ecosystem functions. And particular traits such as diet are used as proxies for their functional roles. So diet can influence trophic dynamics, energy flow, ecosystem processes like herbivory, and body size is another commonly used trait, as it can provide information on feeding function, movement, home ranges, and energetic needs. So using these traits, we can create measures of functional diversity, and these can inform the likely outcomes or possible outcomes of shifts. So within this, we can position species according to their trait combinations in a multi-dimensional space. And I've just pulled up 2 axes of that functional space here, but actually for my analyses, for 2 of my chapters, I've actually used 4. But species are organized, and as you see, for example, these 3 species might share the same kind of functional traits. And from this, we can get a measure of functional richness, which is the spread of traits, the range of available trait combinations. And in this example, Assemblage 1, for example, would be functionally richer than Assemblage 2 because it covers a wider area of functional space. Functional evenness is another commonly used measure. In this example, circle size is relative to abundance. And so Assemblage 2 would be more functionally even than Assemblage 1. And another commonly used metric is functional divergence, and this is a measure of how specialized an assemblage is. And this is essentially calculated by what the relative abundance is of species sort of towards the periphery of functional space. So for this chapter, as I mentioned, I looked at how functional diversity can vary among different habitats. I used these 6 habitats here, and we collected the fishes and then put all 218 or so fishes into this trait-based analyses, and I considered 6 commonly used traits. So diet, body size, mobility, so the extent to which species move within or across reefs, the time that they're typically active, their social grouping, so whether they're solitary or they're usually found paired or schooling, and the position that they're found in the water column. And from that, I then took measures. I took the measures of functional richness, so the range of trait combinations, the functional evenness, so how even abundance is distributed, and also the functional divergence, so how specialized those assemblages are. And I compared them among all 6 habitats. So what did we find? Well, functional evenness didn't vary among habitats, but functional richness, so the spread of traits, and also functional divergence, so the extent of specialization, did vary among habitats. This here, we've got a plot of functional richness among habitats. And what we found was that branching parietes had a greater functional richness, so a greater range of functional entities than the Poecilopora and soft coral habitats. Similarly, for fish functional divergence, so the specialization, the parietes habitats, so branching parietes and massive parietes, had a greater extent of specialization than Poecilopora, and these relationships were best predicted by this differential habitat structural complexity. So here we've got structural complexity across the x-axis, functional richness on the top and functional divergence on the bottom, and up at this end we have all the branching parietes habitats up here, and the same here for the massive and branching parietes. And at the lower ends, the lower relief soft coral and Poecilopora habitats. So I was interested in seeing which traits were driving these patterns, and this is the original functional space. So this doesn't account for a particular habitat or for the abundance of any of the species. It's just presence, absence, ordered in functional space according to trait values. And from this functional space, we can then see how the habitats vary. So I've just pulled up one, and I'll bring up the rest in a moment, but just so you can see, the circle sizes are relative to abundance of different species. And you can see that in Poecilopora habitat, there is higher relative abundance of sort of sedentary farming damsels and pairing corallivores. If we compare that to the other habitats there, I'll just sort of let you have a look at that. Again, the colours are just to differentiate habitats, and circle sizes are to show abundance. And if we look at where there were significant differences among habitats in the extent of assemblage specialization, you can see that in branching and massive parietes habitats, there's a much higher abundance of these schooling nocturnal planktivorous fishes. So to sum up the findings from this chapter, the functional structure of reef fish assemblages varied among habitats. The range of trait values or functional richness, as well as the abundance of specialized traits, so functional divergence, were mediated by differential structural complexity, with trends driven primarily by these small-bodied schooling planktivores in the parietes habitats. So what this indicates is that these altered coral assemblages may well influence the distribution of fish traits going forward. And this raises the question of what the implications are for the stability of these distinct species assemblages with ongoing disturbance. Which leads me on to my 3rd chapter. So we know that the different coral habitats provide different structures, and we know that these distinct habitats support different fish assemblages. But what happens when a really big bleaching event happens? So I was lucky enough, I suppose, to have data before the really unfortunate bleaching event. And so I was able to, I had data from about 6 or 7 months before the bleaching. In 2016, I went back to Lizard Island during the bleaching event, and then I returned there again some 6 or 7 months later. And I just resurveyed the benthos and the fish assemblages each time so that I could compare them. So I was most interested in what was the extent of pre-disturbance coral species composition on the susceptibility of reef fish assemblages to coral bleaching events, because we know that coral bleaching events, typically where it incurs huge coral loss, will have huge detrimental impacts on reef fish assemblages. But I wanted to look specifically at the role of coral species composition in this. So I compared data from these 6 habitats you can see here. And I took this fish data, I assigned those same traits that I showed you in the previous chapter, and I compared, and all of this is for fishes, by the way. I compared the total abundance of fishes, the species richness, so the number of fish species, functional richness, which, as you'll recall, is the range of trait combinations. This time I looked at functional dispersion, which is a measure of how variable the abundance is of particular traits within an assemblage. I also took a measure of functional originality, which is really an indicator of functional redundancy. So it's calculated as the isolation of species within that functional space. So if there's only one species in a particular part of that functional space, it would have a higher functional originality, and therefore the assemblage would be considered to have a lower functional redundancy. And I also compared the functional structure using community-weighted means for each functional space. And I compared that 7 months before the bleaching in September 2015, during the bleaching in April 2016, and then 6 months later in October 2016. And again, I used a range of mixed effects linear models, and I did that both within time periods among habitats. This will become much clearer when I get to it, but also I looked at changes over time within each habitat. So this is the functional space, and I'm just going to show you this because it's going to be important for some of the results later. This is the functional space that I used to calculate functional diversity. And I use 4 dimensions, and these are the first 2. And just to sort of talk you through what you're seeing, down here the traits cluster with the sort of small-bodied, benthic attached farming damsels and corallivores, moving up to the much larger-bodied piscivorous and omnivorous fish species up there in that top right-hand corner. And over to the left side, we've got the large schools of planktivores. In the 3rd and 4th axes, actually the traits cluster together much more closely. But what I want to draw your attention to, because this is going to be important, is outside of this cluster, we've got these 3 groups. Particularly, we've got these small-bodied farming damsels that are arguably habitat generalists. So using these 4 dimensions of functional space, I was then able to extract measures of functional richness, functional dispersion, so that variability within habitats, within assemblages, as well as functional originality, which is an indicator of functional redundancy. So what did we find? Well, let's first look at what happened to the corals. So we know from the work of Terry and a number of people in this room that there was mass coral bleaching in that northern section of the Great Barrier Reef, and Lizard Island was no exception. Though there was a little bit of variation in bleaching among the habitats, there was no significant differences. If we compare that to the loss of coral that was documented either during the bleaching or after, actually significant coral loss was only found in 3 of these 6 habitats. So in branching Priorities habitats, we found a 15% decrease. Mixed coral habitats, which had a lot of, a range of things by definition, but also a lot of acropora, we found an 18% decrease. And in soft coral, there was a whopping 31% decline in coral cover. So what did this mean for the fish? Well, What you're seeing here, and I'm about to bring up a whole panel of plots, is first of all the species richness of fishes. What you can see here in these black points is in September, so before bleaching, the grey is during bleaching, and the red is after the bleaching. And the habitats are organised along the bottom. And I don't need you to focus on specific values for the habitats, but I'm really happy to talk that through with anyone during question time. But what I wanted to show here was that there was an increased similarity of fish assemblages through time. So, for example, before the bleaching, there were 3 differences among the habitats in species richness. During the bleaching, there were still 3 differences among habitats, but actually the differences changed. By 6 months after the bleaching, there were no differences among habitats and species richness. If we compare that with the other measures I looked at, we find the same trends. So in total abundance, prior to the bleaching, there was quite a high distinction among the habitats with 7 interhabitat differences. During the bleaching, down to 2, and after the bleaching, just 1, with branching parietes having significantly higher total abundance of fishes than degraded reefs. Functional richness didn't change, so there was no loss of functional entities, indicating that actually it was just changes in local abundance rather than entire loss of species, or functional entities rather. Functional dispersion, so the variability, went from 3 differences down to 1, and this was due to a loss of to lower abundance of particular specialists in branching parietes habitats that hiked up the overall levels by the time we got to after the bleaching. And the same for functional originality. We went from 7 interhabitat differences before the bleaching to none, and again this was due to some changes in branching parietes habitats. So you might wonder, well, which traits are driving these shifts? And what I've shown you here is, you might recognize them, these again are the functional spaces that are relative to each of the habitats, which are listed across the top. And they refer to each time period. So we've got before the bleaching on the top row, during the bleaching on the second row, and after the bleaching on the third row. And what I'm showing you here is the third and fourth dimension. The first and second dimensions of functional space are really consumed. Most of the variance is explained by differences in body size and diet. And whilst those traits didn't really change, what we're picking up here is a really subtle but significant shift upwards in functional space across all of these habitats in the direction of these small-bodied generalist farming damselfishes. So the second main finding for this chapter was that there was an overall increase in functional originality at the system level, which indicating a potential loss of functional redundancy. And as I mentioned before, this was due to an increase in functional originality, or rather the loss of or decline in abundance of particular specialist live coral specialist species in the branching parietes habitats. And what I found actually really interesting was that coral species composition was a better predictor of changes in the fish assemblages than the extent of coral loss. What you can see here is the 3 habitats where there were declines in coral cover. In branching parietes, the fish assemblages were arguably worse affected, with declines in species richness, increase in the variability of traits of functional dispersion, and an increase in functional originality, so a loss of functional redundancy. In mixed coral assemblages that incurred a greater loss of coral, there was no change detected in the fish assemblages whatsoever. In soft coral, which incurred a 30% loss of coral cover, the only detected change in the fish assemblages in those habitats was actually an increase in total abundance of fishes. In Posilopora, where there was no change in coral cover, there was a decline in species richness and functional richness. And in massive parietes, despite comparable bleaching, there was no change in either coral cover or in the fish assemblages. So just to summarize this chapter, there were differential responses of corals and fishes to severe heat stress, which caused an increased similarity of fish assemblages or biotic homogenization, both functional and taxonomic, with a potential loss of functional redundancy. The changes in fish assemblages were better predicted by pre-bleaching coral composition than the extent of coral loss. And this really highlights the role of coral species composition in both structuring communities as well as predicting their responses to disturbance, such as a large bleaching event like this. Which raises the question, what is the capacity of these emerging configurations to maintain critical ecosystem functions? Well, this is my last chapter, so if you're still awake, well done. I asked whether critical herbivory functions vary among distinct coral habitats, and where the previous chapters were published, I'm just going to point out that this one I'm still working on. So herbivory, as you all know, is a really critical process on coral reefs, and it's been linked to the resilience of reefs as herbivore species can remove algae which would otherwise compete with corals for space and limit recruitment of corals. So evidence thus far shows us that herbivory can be highly spatially variable. The distribution of herbivores has been shown to vary with depth and reef zonation, both in terms of their abundance as well as their biomass. The distribution of herbivores also can be highly variable with reef condition as well as differential structural complexity. If we look at rates of herbivory, these have also been shown to vary, and we're going to sort of narrow down in scope here. Herbivory has been shown to vary according to geographic position or position on the continental shelf. Similarly, this is a figure from that same study I showed you just now. Herbivory rates can vary with reef zonation. But studies have also shown us that herbivory rates can really vary within habitats, and it's been speculated that this is due to differences in benthic composition, possibly due to differences in coral cover or structural complexity. And this is what I'm most interested in. We don't yet know what the influence is of coral species composition on important rates of herbivory. So that's exactly what I looked at. I compared herbivory among 3 habitats. And again, I took measures of estimates of the herbivore assemblages at each site. And I compared both browsing and grazing. To compare browsing, I used Laurentia obtusa assays, and I had 6 exposed to herbivores at each site, as well as paired controls. And I left them out for a period of 24 hours on the reef. And I filmed them to see who was responsible for eating them. I took the wet weight before and I compared it to the wet weight after. For grazing, I used turf assays that were preconditioned with lovely similar turf assays with a similar height of turf. And I put down again 6 assays onto each site with cage controls, and I left them out for 7 days and compared the mean height before versus after. I then used again a range of mixed effects models, and as well as a bit of model selection to see what was best predicting the patterns that I found. So whilst there was no variation in grazing among habitats, grazing was evident, I should mention, among habitats, but there was no variation between them. There was a variation in browsing among habitats. So there was no significant differences in the cage treatments here in the black. Here we have the loss of Laurentia obtusa. But there was a significant difference between those cage controls and those assays that were exposed to herbivores in the mixed coral and soft coral assemblages, though there wasn't a significant difference between these. Interestingly, reef condition was the best predictor of browsing. So many of the studies that have come out so far have speculated that higher structural complexity or higher coral cover can be correlated with higher rates of herbivory. In fact, it's not really a speculation, that's what they found. I've actually found the opposite. So with increasing cover of dead coral and algae, we found a greater loss of algae from these macro assays. And I've actually just plotted the raw data here just to show you that the caged assays don't change at all on that gradient, but there isn't a steep incline on the exposed ones. The cover of dead coral and algae was actually highly collinear with coral cover. So using that, we can then plot it with total coral cover, and of course, it's the mirror image. And you can see that as with an increase in total coral cover, you have less removal of macroalgae. What was also interesting was that the removal of macroalgae from the assays didn't correlate with the visual estimates of browser biomass. And I'm going to explain a bit more about that now. So after watching a few hours of videos, I was delighted to see that reef fish really like Laurentia. And we found, I counted 36 different species, but only 4 were responsible for more than 95% of the mass standardized bites. So they were the most responsible for that loss that we found, at least during the hours that were filmed. If we look at the bite rates, as you would expect, it mirrors the pattern we saw for the Laurentia loss. Branching parietes is significantly less than soft coral, and there was some variation in the mixed habitat, primarily because they got smashed by these guys. But anyway, if we compare that, if we look at the bite rates by these individual species, so we had a couple of nasos here, the Pomacanthus sextriatus and Siganus doliatus. What was interesting was that the angelfish was the only one found feeding in the branching parietes habitats, and actually its feeding activity was consistent across all 3. But feeding by the other species was absent in those habitats. Now, is that because those species weren't there? Well, if we compare that with the visual estimates of both all of the nominal browsers put together, as well as that of each of the different 4 species, you can see that actually there's really not a lot of correlation here at all. In fact, this naso was completely absent from the visual census. So this isn't new. With numerous studies, including those done by Andy and others, have shown that transect surveys of fishes can be poor indicators of the functions being carried out. But it's just interesting that these findings echo those results. So just to sum this up, contrary to expectation, reef condition best predicted browsing, as it was negatively correlated with coral cover. Browsing varied among these coral habitats, but it didn't correlate with visual estimates of browser biomass. And grazing herbivory didn't vary among habitats, but interesting, neither did the visual estimates of grazer biomass. So, we're now coming to a close. I'm going to sum up the main findings of each of the chapters and then sort of have a discussion about what this might all mean, and then you're free to leave. The first chapter, we found that structural complexity varied among habitats, but some habitats provided complexity across more scales than others, which has important implications for resilience. The functional structure of reef fish assemblages varied among habitats, and these differences were best predicted by differential structural complexity. And those differences were driven by the relative abundance of small-bodied planktivores in the highly complex habitats. Chapter 3 showed that species turnover with coral bleaching increased the similarity of reef fish assemblages and indicated a potential loss of functional redundancy, with changes in the fish assemblages better predicted by pre-bleaching coral composition than by the extent of coral loss. And finally, I showed that browsing processes varied among habitats, and this was best predicted by differential reef condition, but grazing processes didn't vary. And this just highlights that there are differential mechanisms going on the reef that are influencing these distinct functions. So what does this all mean? Well, the findings really illustrate the role of coral species composition in shaping the physical structure of habitats, in mediating the structure and function of reef fish assemblages, as well as influencing the ecological responses of reef species communities to disturbance. As we move forward into the future, and we're seeing, we're going to see more and more of these species shifts, and we're going to get some novel reef species configurations. I guess these findings fit into this in that we're likely to see a more structurally homogenized reef environment, though there's going to be differences between the habitats depending on the outcome of the species shifts. This is going to have an influence on the distribution of particular fish traits, especially body size and diet, and the extent of specialization. What we see with disturbance is due to the differential trait susceptibility, we're going to probably see more biotic homogenization as specialists are lost, and we have greater abundance of generalists in their place. And this indicates that we may have compromised functional redundancy as well as resilience, which really all comes together to indicate that we don't know really the capacity of these coral reefs of the future for providing these essential goods and services for the communities that depend upon them. And with that, I want to I just pulled out this quote because I think it really summarizes the importance of community composition in influencing ecosystem structure and function. A paper by Kent Redford called The Empty Forest said, "We must not let a forest full of trees fool us into believing that all is well." Here is some of the papers from my thesis. As I mentioned, this last one is in prep. I'm hoping to submit it really soon. And with that, I just want to extend an enormous thank you. Of course, I didn't do this all by myself. My supervisors are amazing. I feel really privileged to have worked with them, and I'm really so grateful for their expertise and their patience and their mentorship throughout all of this. I was assisted by a lot of people in the field, particularly Jacob, who counted a lot of fish. A number of people helped me to learn some really complex stats. My friends are incredible and have kept me sane and balanced throughout all of this, most of the time. And of course, the Centre has been really generous in funding my project and supporting me, and I feel very privileged to have been here. I've also received a number of research grants and travel grants, and I just want to acknowledge them as well.